Welcome, everyone, to the Society for Developmental Biology's postdoctoral seminar series. We are very excited to highlight the work of our outstanding postdoctoral members. Today, Alys Jarvela from the University of Maryland and Ryan O'Neill from the NIH will share their research. Each speaker is going to give a 20-minute presentation, followed by 10 minutes of question and answers. If you have questions, please enter them in the Zoom question and answer box. Additionally, make sure you stay until the very end of today's seminar after the second talk, because we've got a special announcement after the talks today. So it is my distinct pleasure to introduce Alys Jarvela, our first speaker from Leslie Pick's lab at the University of Maryland. And it's particularly fun for me to introduce Alys because we both studied echinoderms for our PhD work, which is a small community. And so it's been really fun to get back in touch and catch up. Alys has always been interested in how developmental gene regulatory networks evolve. And she's worked on that both as a PhD student in Veronica Hinman's lab and now also as a postdoc. So her work has covered a variety of topics in that field. As a graduate student, she studied both how an individual transcription factor's preference for a particular binding motif can change, which I think is really interesting because typically I've always thought of how the motif changes in the genome rather than how the transcription factor can change. And also she studied how the gene regulatory network governing neural ectoderm patterning functions and how that might then change through evolution to generate different numbers or diversity of neurons. She switched to insects for her postdoctoral work, and she's focused on the mosquito. And today she's going to tell us about her studies, which have shown that there's been a switch in the developmental genes that are controlling segmentation in the mosquito. So it's really exciting. I'm looking forward to your talk. She's also very interested in insect conservation. She does a lot of native plant gardening, and she works with the University of Maryland Bug Zoo, which I think is really cool. And you can correct me if I'm wrong, but I think this is kind of like this amazing sort of outreach program where you can like pet tarantulas and see beehives and scorpions and things like that. So that sounds really exciting. Unfortunately, she also says that her 5-year-old has taken that to mean that it's okay to have ants in the kitchen. And as a fellow parent of elementary school students, I totally empathize with you. So we're looking forward to a great talk. Take it away.
Thanks, Dan, for that really nice introduction. As he mentioned, I've been interested in how gene regulatory networks develop throughout my career. And today I'm going to tell you about how we took advantage of the vast, rich knowledge we have in Drosophila and applied that to a non-model fly, which is the mosquito Anopheles stevensii. And so the mechanism underlying segmentation is really, really well known in Drosophila. All insects have a segmented body plan, and it starts with maternal effect genes, which are expressed along the anterior to posterior axis. Those activate the expression or regulate the expression of gap genes in broad swaths across the anterior to posterior axis. Those then regulate the parallel genes such that they're typically expressed in 7 parallel stripes. And these stripes prefigure the primordia of alternating segments. These will be the segments in the larvae that will eventually come from this embryo. The parallel genes regulate the expression of the segment polarity genes. Those are expressed in each segment, and they determine whether a portion of the segment will be the anterior or the posterior. In Leslie Pick's lab, where I'm a postdoc, we focus on this last step of the network, understanding how the parallel genes regulate the segment polarity genes and how that process changes or is conserved across different insects. Our lab and many other labs have started to look at this network in a vast array of arthropods now. And so what we know from looking at all these different arthropod species and looking at how they generate their segments over development is that there is a gene called paired, which is essential for segment patterning in a lot of different arthropod species. And just to really simplify it and just show you the basics, starting with what we know in Drosophila, there's a segment polarity gene called engrailed, which is expressed in 14 stripes in the embryo. And then this corresponds to how segments are patterned in the larvae. You can see that each segment has a part of smooth cuticle and some bumpy cuticle called the denticles. Now, when you have a mutation in the paired gene, every other engrailed stripe is lost in the embryo, and every other morphological segment is missing in the resulting larvae. And this effect is lethal. When we look at how this occurs during beetle development, it's a really similar situation where if you knock down paired using RNAi, every other engrailed stripe is greatly reduced compared to the control, and every other morphological segment is missing from the resulting larvae compared to a wild-type larvae. And again, this is lethal. In the mosquito, we see a really normal conserved expression pattern for engrailed in the embryo. We see a really normal larval body plan with 3 thoracic and 10 abdominal segments. The sort of strange thing about this is that this is already the paired minus situation because paired is not present in the genomes of mosquitoes. And so if you want to understand how you can lose a gene that's required for viability in other insects, but has no morphological consequence in the mosquito, our first step was to really verify that this loss had occurred and try to identify any potential match to this gene in mosquito genomes. And so as a first step, I did some reciprocal blast searches and some Hummer searches and all of the available mosquito genomes. And I built this gene tree using those results. The hits from the mosquito genomes are shown in bold. And so paired is a PAX37 transcription factor. And so all of the different PAX3 transcription factors are sort of in this larger clade here. We know that we weren't overly stringent in our search because we also recovered matches to PAX6, which is also known as Alys Jarvela. And you can see that there are mosquito hits in all of these. Now, all of the PAX37 genes we identified either group with gooseberry or gooseberry neuro. None of them go into this paired clade where we just have our reference sequences from Drosophila, from flower beetles, and so on. Another way we can further confirm the identity of these candidate matches is through signature sequence motifs. It's known that in other insects, gooseberry and gooseberry neuro have this highly conserved octopeptide motif. You can see there's really good alignment here, and then it's more specific to each gene on the N-terminal end. Paired genes have no alignment within the equivalent region, and so the mosquito genes shown in bold always belong to either gooseberry or gooseberry neuro based on the presence of this motif. We also know that these genes are gooseberry and gooseberry neuro based on synteny, so looking at where they are in the genome. Orthologous genes are shown in equivalent color here, and gooseberry neuro are almost always adjacent, or sometimes they're just very nearly adjacent. There's really good conservation of their directionality with respect to each other. And then if you focus on the diptera, which are the flies, you also see high conservation of the genes immediately surrounding gooseberry and gooseberry neuro. And so that bolsters our confidence that we've identified these genes correctly. In contrast, if we look at the synteny around paired, there's really essentially no synteny, even at really recent divergence times. If we look within diptera, even if you compare Drosophila to, say, the housefly, there's no synteny around paired. When we look at these 3 species, which are the closest relatives of mosquito that do have paired, there's no synteny around paired here either. That was a little disappointing because it means that we can't determine where paired ought to have been in mosquito genomes to see if there's a pseudogene. But it does suggest that there's a lot of rearrangement here, or maybe some transposable element activity, and that's how paired was possibly lost in the first place. So to summarize what we know so far, mosquito genomes have 2 PAX37 family members. We know this based on phylogenetics, signature sequence motifs, and synteny. And the 2 genes that they do have are gooseberry and gooseberry neuro. The gene that was lost was paired. Now, something that we know about paired and gooseberry is that they are transcription factor paralogs that arose from a gene duplication event, and that duplication occurred in the crustacea or possibly earlier. So before insects existed, paired and gooseberry existed, and they've been diverging ever since. So in spite of the fact that they've been diverging for 500 million years, Their molecular functions are still essentially the same. There was a nice set of experiments done by Marcus Knoll's lab a number of years ago. Just to remind you, when paired is wild type, you have 14 stripes of engrailed. When you lose paired, you lose alternate engrailed stripes. And then what the Knoll lab did is they developed a transgene where the gooseberry coding sequence was driven by the paired cis regulatory elements. And when they provide this transgene to paired mutants, they're able to recover those lost engrailed stripes. So what this means is that gooseberry is capable of performing the functions of paired as long as it's present in the right places and at the right times. And so this led us to think about a possible mechanism for how paired was lost in mosquitoes and more generally how genes could be lost. If you have a scenario where you have a transcription factor and its pair log is able to regulate its target genes, then you should be able to lose that gene without disrupting the larger gene regulatory network. And so we took what we knew about paired in Drosophila and we developed some criteria for testing whether Anopheles gooseberry has taken its place. So in Drosophila, we know that paired is expressed in a parallel pattern at the blastoderm stage. We know that it activates expression of engrailed. And we know that alternate segments are missing in the mutant larvae. And so we would expect that each of these three things should apply to gooseberry and mosquitoes if gooseberry has taken the place of paired. We first looked at gene expression by in situ hybridization, and this was performed by an honors thesis undergraduate, Katie Trelstead, who worked with me on this project for quite a few years. Just to show you for reference, we first looked at Drosophila, and we know that paired is expressed first as a single stripe and then as 7 parallel stripes. At the gastrula stage, it briefly takes on a 14-stripe pattern. It activates gooseberry, and then paired turns off, and gooseberry maintains the 14-stripe pattern through the next phase of development. It's important to note that gooseberry is not expressed during parallel patterning. When we look at how gooseberry is expressed in Anopheles, we see a merger of these two patterns, where we see in the blastoderm stage a paired-like expression pattern of broad stripes, but we also see conservation of this later 14-stripe segment polarity pattern. And so this suggested to us that the functions that are divided among these two genes in Drosophila could all be performed by this single gene in the mosquito. We also see that gooseberry is expressed in the correct register to be performing paired's function. So what I mean by that is that in Drosophila, paired regulates the expression of the odd-numbered engrailed stripes, and a different parallel gene, FUTS, which is shown in magenta here, regulates the expression of the even engrailed stripes. And paired and FUTS are expressed in this alternating pattern with respect to each other. And we see a conservation of that alternating pattern when we look at how gooseberry and FUTS are expressed in the Anopheles embryo. We next wanted to study Anopheles gooseberry function, and to do this, we used Cas9 and homology-directed repair. What we did is we replaced the first exon of gooseberry with a 3xp3 GFP cassette. What this does is it labels insects that have the mutation. Adults have fluorescent green eyes and larvae have a fluorescent nervous system. This is important because we predicted, and this turned out to be true, that homozygous mutants would be lethal. So we needed to have a mechanism to maintain heterozygotes, and there are no balancer chromosomes in Anopheles like there are in Drosophila to keep track of those types of mutations. And so in order to obtain homozygous mutant embryos, we set up crosses where both parents were heterozygous for this GFP knock-in into the gooseberry gene. And as a control cross, we used GFP-positive females just in case our CRISPR introduced any unpredicted maternal effects, and we crossed those to their wild-type male siblings. Now, when we look at the embryos that are progeny of these crosses, control cross embryos always have a wild-type engrailed expression pattern of 14 stripes. But our gooseberry cross, where we would expect 25% of the embryos to be homozygous mutant for gooseberry, we see that 25% of the embryos are missing alternate engrailed stripes. And that indicates that in this species, gooseberry activates engrailed. When we look at larvae that come from these crosses, we see that in the control cross, all larvae are wild type. So that means that they have 3 thoracic and 10 abdominal segments. And we see that in our gooseberry cross, 25% of the larvae look like this, where they only have 2 thoracic and 5 abdominal segments. We can tell that in these gooseberry mutants, it's always the same set of segments that are missing. We can tell them apart based on some landmarks, like T2 always has 2 pairs of setae, these long hair-like projections. So that's missing here. Also, A9 is a highly derived segment. It's a respiratory siphon that's always missing in our gooseberry mutants. And so it's always alternate segments that are missing. And this looks strikingly similar to what the parallel mutant for paired looks like in Drosophila compared to the wild-type cuticle. So to summarize, for gooseberry and Anopheles, we predicted that we would see parallel expression if it was performing the function of paired, and we do see that at the blastoderm stage, but we also see a maintenance of the segmental expression later on. We see positive regulation of engrailed, and we see loss of alternate segments in the mutant larvae. So this led us to propose an evolutionary scenario about how paired and gooseberry subfunctionalize over time. We know from a variety of insect species that paired is usually expressed earlier in development and performs the earlier developmental functions. Gooseberry is expressed later and performs the later developmental functions. And then this state was inherited by all of these different insect lineages. And then specifically in the mosquito lineage, what we think happened is first gooseberry acquired this early parallel type pattern that caused it to be redundant with paired early, but later only gooseberry was available to perform important developmental functions, and therefore paired was the gene that was lost. Many years ago, Ina Gittleman coined this process synfunctionalization. So that means that you've reversed subfunctionalization, you've remerged functions that were divided among 2 paralogs into a single gene with all of the functions. And we think that this is probably a really common mechanism of gene loss that could be seen in many different organisms, but it's really hard to show for certain that it occurred because you need to know that a gene was lost, you need to know what that gene's function had been with evolutionary context, but likely it's prevalent but understudied. This work also led us to develop a hypothesis about how cis regulatory elements will evolve or not evolve in response to this process. And so in Drosophila, again, we know the whole gene network for segmentation extremely well. And now we've developed this Anopheles system to compare it to. And so if we think about the cis regulatory element for engrailed in Drosophila, it might look something like this, where there's a piece of DNA upstream or downstream of the engrailed coding sequence that has binding sites for all of the different known regulators. One of them is paired. And so what's happening in the mosquito lineage is that gooseberry is just binding to the binding sites that used to be recognized by paired. Nothing functional about this piece of regulatory DNA will have needed to change. And so that's why this process would work, is because you wouldn't have to change all of the target gene CREs. And so if this is true, we should be able to find this region of DNA in the Anopheles genome, put it into transgenic Drosophila, and it should be able to function. It should be able to produce an expression pattern that looks like engrailed. And so this is exactly what we've been working on lately. We've started by identifying candidate regions in the Anopheles genome, putting those into reporter constructs. We're then developing transgenic lines in Drosophila, and then assessing reporter gene expression and seeing if we can find regions that recapitulate engrailed-like expression in Drosophila. We started doing this by looking at sequence conservation using a program called EvoPrinter. What that does is it takes advantage of the fact that there are many, many sequenced Anopheles genomes, and you can use sequence conservation then to predict candidate regulatory regions. So here are where a few of ours are with respect to the engrailed gene. And then we put these into transgenic Drosophila. We looked at gene expression. And what we saw was, while each of these directs some known aspect of engrailed expression, some of them weren't really relevant to the developmental time we were interested in. Some of them were sort of weak. And then I think this one's also maybe a little bit later. This is already sort of mesodermal and maybe not directed by parallel genes. And so then we did a FAIR-seq experiment to look for candidate regions where there is open chromatin at the right developmental time to be relevant to our study. And so here's what it looks like for engrailed. We got a really strong fair peak right upstream of engrailed. And this overlaps with one of the regions we've tested, but suggests that maybe we get a better result if we expanded that region out a little bit. We're also finding other candidates far upstream of the gene that we hadn't considered before that we're going to test now. And the great thing about this data set is that it can be applied to look for regulatory regions for other Anopheles gooseberry target genes. We can use it to study how gooseberry acquired its early expression pattern in Anopheles. And so we're hoping to do a lot with that in the future. The other thing we're doing moving forward is trying to produce transgenic Anopheles using these cis-regulatory reporters. And so just like in Drosophila, we're finding these candidate regions, we're focusing in on the ones that worked well in Drosophila. We're producing transgenic Anopheles lines. Notably, this is more time-consuming, challenging, and expensive in Anopheles. That's why we started with Drosophila, even though it's the cross-species experiment. And then finally, we're going to try to assess reporter gene expression once we have those lines. So I'd like to thank the PIC Lab for their ideas and support throughout this project. I really like to thank my PI, Leslie Pick. I started this totally new species in her lab in this new project, and she was really patient with me. Also, Katie Trolstead, again, who was an undergraduate who worked on this project and was second author on our paper. Everyone in this lab has helped me at some point or another caring for the mosquito colony. They are very labor intensive. We also got a lot of help from mosquito experts within our department, like Megan Fritz at the Insect Transformation Facility at the University of Maryland, Shady Grove. They helped us really learn the ropes of using the mosquito systems, caring for these animals. And ITF was also really important for producing our CRISPR line in Anopheles to do our functional experiments. Our work was funded by the NIH, and thank you for listening.
That's great. Thank you so much for a wonderful talk. I would like to remind everyone listening that if you have a question, you should type it into the question and answer box, and then we will answer it live. I'll get us started. I just wondered if you could comment at all about why it might be useful to lose paired and gain gooseberry, and are there other functions of paired and gooseberry that are different between mosquitoes and other insects that might suggest like sort of the why of this?
I don't know about the why, but one possible consequence, I know another function of paired in Drosophila is it's important for the male accessory gland. And I think in that no lab experiment that I described earlier, they saw that paired and gooseberry weren't redundant there. And so I really don't understand what happened in that case, but I know that like sex-specific things turn over more rapidly in evolution. Maybe that promoted divergence. Maybe there's something really different happening in mosquitoes versus Drosophila that could be a target for pest control. That there could be something interesting there, but that's sort of way outside my wheelhouse.
Cool. And then another, hold on, a question in the question and answer box. Okay, first question, very interesting. This is from Zainab Afzal, very interesting. What could you change in Drosophila gooseberry for it to act like paired? I guess I'm trying to understand what additional features mosquito gooseberry gain to be able to bind paired sites.
That's the thing. We don't think it gained anything at the protein level, because in Drosophila, they already showed that you could interchange these two proteins. And at least in the case of segmentation, it doesn't matter. A lot of genes do this. They'll duplicate, and then instead of the two duplicates taking on different protein sequence and different molecular functions, they just develop different gene expression domains. And so the only thing that seems to be different about paired and gooseberry and Drosophila is when they are expressed. And so all that had to happen in mosquito was a gain of that early expression pattern, which is probably why it was able to happen. It's a pretty simple change.
Do you think that you could you express the mosquito gooseberry in a fly and see if it rescues the paired? Like
I mean, I would expect that it would. We haven't done that experiment, but I don't see why that wouldn't be true.
Cool.
but I have no idea. It'd be interesting to see if it rescues those other functions. Again, Drosophila gooseberry can't rescue other functions of Drosophila paired, but maybe mosquito gooseberry could. We don't know that.
Yeah, so if you expressed it from the Drosophila paired regulatory element, you might be able to detect if the protein actually really has all the same function. Okay, we have another question. This is from Eric Clark. Thanks for the very nice talk. Do you know what paired or gooseberry gene presence expression looks like across other dipterans? And does the Anopheles gooseberry mutant show any segment polarity defects in addition to the pair rule phenotype?
We didn't see segment polarity phenotypes in the mosquito, but I know in Drosophila, a lot of times those are sort of covered up by the like the previous pair, because the pair rule phenotype kind of occurs first, and then it sort of obscures the downstream segment polarity phenotype. I know that in Drosophila, you need some kind of special lines to parse those two things apart. A lot of pair rule genes also have a segment polarity function, so we don't know that in the mosquito. We would expect that we might see conservation of regulation of wingless and some of those other segment polarity targets that gooseberry would ordinarily have, but we haven't actually checked that. The other part of your question was about expression in other diptera. Diptera. I don't think that's been done. I know that there was somebody had done CRISPR in the Mediterranean fruit fly or something similar, where they used paired as like their control situation, and they knocked down paired and they got a pairal phenotype. So that's predictive of conservation of that function, but we don't know anything about the expression.
Okay, sure. Insert. Yeah. While we're waiting to see if anyone else wants to ask any questions, you showed sort of a diagram of the gene regulatory network, and I didn't really get to sort of like pick it all apart, but like how much do you know about how this sort of is all fitting together in sort of a GRN functional unit, like analogous to what's thought about in the sea urchin, sea star, like in the mosquito. Do you have that kind of knowledge as well that you can sort of like specifically inquire?
I know what you mean. I feel like say like the skeleton network in the sea urchin is maybe a little more extensive in terms of understanding like the eventual functional target genes. Like here are the genes that encode the proteins that make the minerals or make the spicual proteins. I don't know that we've gotten to that level in Drosophila. There's some knowledge about like maybe toll genes being the ultimate thing that makes the segments, but that's not really as well known.
I guess I was thinking
in terms of like just the interactions between the upstream parts of the network and downstream. I think that's equally well known as in the sea urchin.
Okay.
It's really hard to make these networks because in the sea urchin, it's like you have this group of cells and they all are kind of using this network. In Drosophila, if you look at like the lines of the segments, like each row of cells is doing a slightly different thing. They're not all equal. It's like they all have their own, version of the network ultimately running. And so it's really, really complicated to draw it in that way and think about it that way.
Okay, one more question. It's about two more questions here. First question from Mark Halfin. Great talk. In Drosophila, engrailed INV is a complex regulatory locus with many enhancers and overlapping functions. It looks like you had a few fair peaks. Do you think there's a similar regulatory structure here? A simpler regulatory structure?
Probably. I know what you mean, like engrailed and invected, they seem like they're highly redundant. I'm sure that we might be seeing some of that. I haven't actually looked to see like where the fair peaks are with respect to one versus the other.
Okay. Another question. This one from Siquian Feng. Following the first question, I feel it would be critical to replace the endogenous paired ORF with the gooseberry ORF and see if such a strain can still finish embryogenesis normally. Your thoughts?
Replace it in I'm not sure I really understand the question, although you just have the text, so.
I do only have the text, so
Yeah. Replace paired with gooseberry in the mosquito?
I would think that means the mosquito, yes. But if you want to type in a follow-up question.
Yeah, I'll think about that one for a second and try to get back to it in the Q&A.
Okay. So next question from Maria Espinosa. Very good talk. I feel grateful for being able to listen to it, and thanks so much for the invitation. So thank you for that comment. I don't see any clarification about the first question, so maybe we'll leave it there. Thank you. It's about 3:30, I think. Yeah, 3:31. So thank you so much. Great talk. And
thank you, and thanks everyone for listening.
Yep.
Great. Yeah, thank you, Alys. It's my pleasure to introduce our next speaker, Ryan O'Neill. Before starting his academic career, Ryan grew up on a dairy farm in New Brunswick, Canada. He did his PhD studying evolutionary genetics with Denise Clark at the University of New Brunswick, and he's currently studying cell and developmental biology at the NIH with Nasser Russan. We're looking forward to hearing about your work about DNA repair and brain size. So take it away, Ryan.
Oh, thanks, Katie. So I just also want to say it's my pleasure to sort of close out this series for the year. I think everyone would like to give a lot of appreciation to the organizers because it's been a pretty cool series, and I hope that we get to see it again next year. So yeah, I'm just going to tell you a little bit about my research. I like to start off with this slide because I think it just shows the incredible complexity of the animal brain, whether you're looking at the massive human brain on the left or the tiny fruit fly brain on the right. It's clear that the proper neuron number, size, and structure of the brain are absolutely critical for the proper functioning of that brain. And I'm really interested in trying to understand what mechanisms control this size and structure of the brain. And so to try to get at this question, I've been investigating a series of human disorders called microcephaly disorders. And in humans, microcephaly is basically when the brain fails to develop to the proper size during development. There are fewer neurons, and it primarily affects the cerebral cortex. Now, over the years, there have been many different genes that have been linked to microcephaly in human patients, shown here on the right. And one interesting thing about these genes is that if you sort of broadly categorize them based on the different functional categories that they belong to, it's clear that there's a lot of centrosome and mitotic spindle related functions encoded by these genes. And also maybe a smaller group, there are some DNA damage repair, DNA damage response functions encoded here. And so these seem to be extremely important processes for determining proper brain size. And a lot is known about how these different genes function in a general sense, but in many cases, there just aren't very many studies, or maybe no studies at all, of how these genes function in the developing animal brain. And so that's really what I've been trying to address, to understand how these genes control brain size using Drosophila as a model. And so we have, the Drosophila orthologs of all these genes here. And my sort of simplistic idea going into this project was we're going to get the Drosophila mutants for these microcephaly genes. We're going to assess the adult brain structure to look for any structural defects in the brain. And then if we find any structural defects there, then we can track backwards in development, look at the actual cells, and try to understand what the heck is going on to actually cause these brain defects. And for most of the talk, I'm going to be talking about this particular microcephaly gene here called TRAPE. And TRAPE encodes a ring domain containing E3 ubiquitin ligase that has multiple known functions in DNA damage response. So first, it has sort of a general interphase role. So in interphase, TRAPE localizes in the nucleus of the cell. It's primarily localized in the nucleolus. And upon DNA damage, TRAPE is released and it can localize to that site of DNA damage where it helps the repair process by recruiting other factors that actually do the repair. And another function that was discovered more recently for TRAPE, so a lot of times cells can, as they're about to enter mitosis, they haven't completely finished genome replication. So in this case here, this cell has an interstrand crosslink that's basically blocking the replication machinery from completing its job. And so what was discovered is that TRAPE ubiquitinates this replication machinery and removes it from the DNA, and that removal is required for other factors to then come in and resolve the sister chromatids so that they can properly segregate. Now, in humans, TRAPE, when it's mutated, it causes a severe form of microcephaly called Seckle syndrome. And in this case, there's actually extreme reduction in brain size, as well as some reduction in body sizes. But given these multiple functions for TRAPE, we really want to understand, what is the mechanism underlying this TRAPE mutant microcephaly. And so to start getting at this question, the first thing that I looked at is, do Drosophila TRAPE mutants have any defects in the structure of their brains? And they do have a structural defect, otherwise I wouldn't be talking. Okay, so here's the Drosophila brain on the left. And if you just look at this, these L-shaped structures in magenta stained with FAST2 or zoomed in here, you can see in the control, these mushroom body structures look like nice L shapes. They're nice and fat. In the TRAPE mutant, they're skinny, reduced in size, and they have structural defects. And you can see the quantification here. And so these seem to be the only structure that was really severely distorted in the TRAPE mutants. And these mushroom bodies, just for some background information, they're really important for learning, memory, and other higher order functions in the fly. And if I take this fly brain and then rotate it and project the 3D volume, you can see actually now labeled with GFP, you can see the cell bodies of the mushroom body neurons called Kenyan cells sitting here on the back of the head. And they basically project their axons forward to form these lobes that we're looking at up here. So if we look at these patches of cells, you can see the TRAPE mutants have a smaller patch of these Kenyan cells. And if we actually count the number of cells, you can see that the number is also reduced there. And some other data that I don't have time to show suggested that this has some characteristics similar with human microcephaly. And so we thought this would be a good model to use for investigating further. So one question I wanted to get at was, what cell type does TRAPE actually function in? And so here's a couple experiments exploring that. I used CRISPR to endogenously tag the TRAPE locus with mneon green. And here you can see the expression pattern of the TRAPE protein in the larval brain. And I'll just point out, these big cells in the brain are the central brain neuroblasts. And that's one of the main places that TRAPE is expressed. Whereas you can see these dark patches, that's actually where the neurons are, the more mature neurons. I also did some cell type specific rescue experiments to look at, mushroom body size in these various rescue conditions. So here's a TRAPE mutant with the skinny, distorted mushroom bodies, small in size, and expressing GFP TRAPE specifically in the neural stem cells or the neuroblasts. You can see that fully rescues this mushroom body phenotype. And again, expressing it in specifically the mature neurons fails to rescue the phenotype. So this really suggested that TRAPE is functioning in the stem cells, the neuroblasts, and we wanted to look a little bit more closely at those neuroblasts. And so to do this, this is looking at a pupil brain, and you can see the mushroom bodies stained with FAST2 and magenta here. And if I just rotate this brain, look around at the back where those Kenyan cells are sitting, you can actually see that there are 4 mushroom body neuroblasts per hemisphere. And these, so basically every mushroom body comes from just 4 neuroblasts. So here are those neuroblasts again in the control and the TRAPE mutant. You can see that the control always basically has 4. The TRAPE mutant has on average about 2 mushroom body neuroblasts at this stage. And that can be rescued by expressing the GFP tagged TRAPE transgene. So where are these neuroblasts going? What's happening to them? Well, I used this genetic manipulation here to basically reduce the level of caspase activity and therefore lower the threshold for cell death and apoptosis. And you can see in this condition, that actually gives a partial rescue to the neuroblast number. So from about 2 to about 3 per hemisphere in this condition here. And I'll just mention this briefly. I'll come back to this at the end. That this manipulation also partially rescues the mushroom body size phenotype. Okay, so why are these mushroom body neuroblasts dying? Well, one of the functions I told you was that TRAPE basically helps repair DNA damage during interphase. And so I looked at these mushroom body neuroblasts using gamma H2AV to detect double-strand DNA breaks, and then counted the number of puncta per nucleus of the mushroom body neuroblasts. And you can see that basically the TRAPE mutants do not have elevated gamma H2AV levels. And so this, along with some other data, suggested that TRAPE is not really mainly functioning in this interphase DNA repair capacity in these mushroom body neuroblasts. But what I did see when I was looking at these neuroblasts, the mushroom body neuroblasts, is several different nuclear phenotypes. So here, about 15% of the TRAPE mushroom body neuroblasts were either polyploid or multinucleate. So you can see the neuroblast outlined here with lamin staining showing 2 nuclei in this cell. And as well, maybe about 9% had these micronuclei. And so these nuclear phenotypes were interesting because they both pointed to potential defects in mitosis. So we used some high resolution light microscopy to just watch mitosis live and see what's going on. So in the control, here you can see a control cell labeled with GFP tubulin and a histone RFP to label the DNA. And that was just to basically show that chromosome segregation happens normally in the controls. Not too surprising. But in the TRAPE mutants, probably about a quarter of these TRAPE mutant neuroblasts had prominent DNA bridges during anaphase. And so these bridges seem to have at least potentially explain this failure in mitosis or the presence of micronuclei, and suggests that there's a mitotic function for TRAPE. And this was quite interesting as well, because when I was looking at this M-neon green TRAPE CRISPR line, I also noticed that during mitosis, whereas TRAPE is normally in the nucleus during interphase, during mitosis, it goes to the spindle and centrosome. And I still don't exactly know what it's doing there, but this does seem to be important for its function. And it's also interesting because a lot of the other microcephaly genes are centrosome and mitotic spindle related genes. So this is to show that the GFP tagged transgene that's overexpressed in the mutant background basically recapitulates the localization of the M neon green endogenously tagged transgene. So it's in the nucleus and interphase, and it's on the centrosomes and spindle during mitosis. And I already showed you this, but that rescues the TRAPE mushroom body size phenotype. So what I did was I deleted the nuclear localization sequence of TRAPE. And you can see here that now during interphase, TRAPE fails to localize to the nucleus. But during mitosis, it still localizes properly at the centrosome and the spindle. And interestingly, it still rescues the mushroom body phenotype. So that suggests that it's the TRAPE mitotic function that's critical for mushroom body size. And I mentioned that this localization seems to be important. We tried disturbing this localization on the centrosome and spindle by relocalizing it to the mitochondria with a TOM20 tag. So you can see here, now it's absent from the spindle, it's absent from the nucleus during interphase. And in this case, this sort of only gives a semi-rescue of the phenotype of mushroom body size. And so that sort of suggests that, you know, this localization might be important somehow, although we don't quite know how. And so here's a model for how I think TRAPE is functioning in these neural stem cells. So if the DNA is not fully replicated going into mitosis, for example, in this case of a DNA crosslink, TRAPE ubiquitinates and removes this DNA replication machinery, and then that removal is required for proper chromosome segregation. So you get proper cell division, proliferation, and normal brain size. And so in the mutant, because these crosslinks can't be removed, then the DNA bridges persist during mitosis. That could either lead to cytokinesis failure, leading to polyploid or multinucleate neural stem cells, or I didn't show you this, but we think it could also lead to chromosome breakage and leading to aneuploid neural stem cells. That could ultimately trigger cell death of the neural stem cells, leading to fewer neurons in microcephaly. And just to sort of wrap up and talk about some future things that I'm looking at. So this is just to show you again that, you know, TRAPE alone has small mushroom bodies, and by using this genetic technique, we can reduce caspase activity and partially rescue the size of the mushroom body. And so I've been trying to use this as a system to take advantage of Drosophila genetic tools and screen for additional pathways that might be downstream of TRAPE in the microcephaly phenotype. And so this is a combination of a targeted candidate screen that I'm showing some results from here, as well as an unbiased efficiency screen. And you can see I have discovered several pathways that seem to be important for this microcephaly phenotype, at least in the TRAPE mutant. And so then going forward, as I discover these pathways and validate them in the TRAPE mutant, then the next step is to see if they're also in play in some of the other Drosophila microcephaly mutants. And this is quite interesting because I only told you about the mushroom body defects in the TRAPE mutant, but it turns out that many of the other microcephaly mutants in flies have this defect in the same structure of the mushroom body, although you can see the mushroom bodies all look totally different, which is fascinating in itself. And so we're going to test these suppressors in these other microcephaly mutants to see if there are common pathways in microcephaly. As well, I think it's going to be important going forward to figure out whether, you know, when we rescue mushroom body size and increase the number of neurons, do those neurons actually have a function? And so using some behavioral learning memory tests to see if these neurons are actually functional neurons or if they're just dead weight. And so, yeah, I hope what I've shown you today is that we can use Drosophila as a great model to look at brain development on multiple different levels: cell biology, developmental biology, and brain structure, and even behavioral analysis. I think flies are a great model to do this kind of work because we can potentially study many genes in a systematic way, since flies are pretty cheap to keep and make new mutants. And the goal is to see if we can successfully model these human diseases and find potential therapeutic approaches to suppress cell death in microcephaly patients. And with that, I'll just thank everyone in the lab and the light microscopy core. Definitely want to thank again the organizers of this seminar series. And I'll take any questions.
Great, thank you, Ryan, for that excellent talk. We already have quite a few questions in the Q&A box. As a reminder, please use the Q&A to ask any questions. So the first question is from Bogdan Serebrnikov. Does TRAPE mutation specifically affect certain classes or subtypes of Kenyan cells? If yes, why is the effect cell type specific?
So it's sort of, the answer to that is kind of yes and no. So the phenotype, I didn't show you this, but the phenotype gets worse and worse over developmental time. And so the earliest born, say, gamma Kenyan cells, they don't really seem to have much of a phenotype. But if you go later on in development to these alpha beta Kenyan cells, those are the ones with the most of the phenotype. But, you know, I don't think that's necessarily a cell type specific effect. It's more that I think that these rare or small defects are just accumulating over developmental time and eventually causing the neural stem cells to die, rather than, you know, I wouldn't say that TRAPE is specifically required for one kind of neuron versus another. I think it's more general than that.
Great, yeah, thank you. Our second question is from Alyssa Armstrong, who says, nice talk, and asks, do you know if TRAPE is expressed in other stem cell populations in flies? If so, what aspects of cell biology are affected?
So this is the interesting question. TRAPE is basically expressed in all proliferating cells that we've looked at. And this is a more generally interesting question, because the same can be said for most of these microcephaly genes. Like, obviously anything to do with, say, the spindle and the centrosome, they're just expressed everywhere. Why is the brain the only thing that's affected? So yeah, in the fly, the only other real defect that we see is that the TRAPE mutant is a maternal effect lethal. And that's basically just to say that homozygous null mothers, they lay eggs that arrest very early in the first few mitotic divisions. But other than that, everything is fine in these flies. Yeah, so it's an interesting question. Why is the mushroom body the only adult structure that's really affected?
So I guess just my follow-up on that question, you think that TRAPE might also have a role in the syncytial divisions, like at the start of embryogenesis? Yeah,
I mean, so what they found in the first paper looking at TRAPE in flies was that basically they just couldn't get through those first few rapid cell cycles. It doesn't even get to a blastoderm. It's like a handful of nuclei that are full of bridges and, you know, just not looking good at all. Yeah, basically TRAPE is required, I think, during those rapid cell cycles to to make sure that any loci that are not fully replicated can be resolved.
Right. Yeah, that makes sense. Cool. Our next question is from Todd Schoborg, who says, cool imaging. It looks like TRAPE was expressed in all proliferating cells of the larval brain. Why do you think the mushroom body neuroblasts are specifically sensitive to loss of TRAPE? Yeah, so this is a question that I can only speculate on, I guess. But it would make sense that, okay, there's biology of the mushroom body neuroblasts are a little bit different than all the other neuroblasts in the fly brain. So most neuroblasts, they divide during embryogenesis, then they go into this quiescent phase during early larval stages, and they only reactivate in the late larval stages to produce a handful more neurons. On the other hand, the mushroom body neuroblasts, they just divide nonstop throughout development. So they go through many, many more cell cycles than other neuroblasts. And they may also have a faster cell cycle. So I think it's just that they have more chances to accumulate these defects compared to other neuroblasts. That's my speculation, at least. Right, yeah, that's interesting. It does sound like maybe the speed that the cells have to divide may, affect how affected they are by TRAPE defects. Yeah. Okay, we have another question from Maria Espinosa, who says, wouldn't it be necessary to determine if the mechanisms are evolutionarily conserved between arthropods and mammals? Yeah, so I think that's a good point. I mean, ultimately, what we tried to do in the lab was to rescue the fly mutation with the human version of the gene, and that didn't work. So it's a negative result. It doesn't really tell you if TRAPE is doing the same thing in mammals versus arthropods. Yeah, ultimately, I think, given that failure, probably some sort of study in a mammalian system would be required to really show that it's the same thing happening. Okay, while we wait for any other final questions, I'll ask another one. I found it really interesting that you could delete the nuclear localization sequence and like you didn't see the same defects. So I guess do you have any thoughts about what might be recruiting TRAPE to like the spindle at the onset of mitosis? Yeah, we really don't know I don't know what the actual recruiting factor is. What I can say is that in cell culture, if you delete the nuclear localization sequence, GFP TRAPE will localize to just interphase cytoplasmic microtubules. So there's definitely something, it is binding to the microtubules somehow, but we really don't know anything about that. Great, thank you. So at this time, we have no more questions. So I'd like to say thank you to Alys and Ryan for your excellent talks. The seminar has been recorded and will be available on the SDB website starting on Monday. And now we are going to have a special announcement by Jessica Feldman from the SDB Postdoc Seminar Series Working Group. Thank you.
Marcia, are you going to share the slides or I can do it? Great. Thank you. So I just want to say it's been a really wonderful, this is our last talk in this SDB Postdoc Seminar Series, and it's been a really wonderful series. So we just wanted to kind of punctuate the end of the series with a couple announcements. So first, go next slide, Marcia. So first, I just want to say congratulations and thank you to all of our amazing speakers who made this seminar series possible. So every one of you gave really an amazing talk that was inspiring for our audience and really hopefully helped you on your journey in science and especially on your job searches. So again, thank you so much and congratulations to all of you. I also want to highlight, so next slide, a couple of our speakers who will be starting their labs next year. So Duan Farmer, Melissa Labonte, Camila Lopez Anido, and Brandon Carpenter will all be starting labs across the country, as you can see here. And so I want to say congratulations, first and foremost, and also encourage undergrads who are watching this series to reach out for graduate positions, as well as graduate students who are watching this series to reach out for postdoc opportunities. So congratulations, and I hope for more of our speakers, we wish you well on your job searches as well. And so finally, next slide. And so one thing I want to say also before we make our second announcement is I just want to say a really big thank you to the community, the SDB community, who made this a really wonderful and vibrant seminar series. And also thank you so much, as usual, to the SDB staff whose time and effort really made this seminar series run smoothly. And so finally, I want to just announce that we've been calling this series the first annual postdoc seminar series, and we're excited to continue this series next year. But we would like to announce the formal name of the series. So next slide, and the next slide, as the Ethel Brown Harvey Postdoctoral Seminar Series. So I encourage you all to look at Ethel Brown Harvey's work from 1909 from her graduate work at Columbia, where she showed that the transplantation of the hippostome from one hydra into another could induce a secondary axis. And so this was 15 years before the famed Spemann-Mangold experiment that won the Nobel Prize for demonstrating the existence of an organizer. And so we just like to highlight the work of Ethel Brown Harvey here, also as a nice counterpoint to the Hilda Mangold seminar postdoc symposium that's at the annual meeting. And so on that note, I would hope to see all of you at the annual meeting in July. And thank you again so much for making this really wonderful opportunity possible for all of our postdocs. So have a really wonderful weekend and a wonderful summer. And again, see you at the annual meeting.